Assalamu alaikum dear students, hope you all are fine. Welcome to the YouTube channel of Multan Public School and College. This is O Levels 8th Biology 5090 and I am your teacher Ms. Zunera Nadeem. Today we are going to discuss a new topic, enzymes. By the end of this video, you will be able to know that what are enzymes, how enzymes are biological catalysts, and what are the actions of enzymes in terms of log and key hypothesis. Before we know that what are enzymes, we should know that what is a catalyst. A catalyst is a substance which can alter or speed up a chemical reaction without itself being chemically changed at the end of the chemical reaction. For example, potassium chlorate. When it is heated strongly in a test tube, oxygen is given off, but this reaction occurs slowly and requires a high temperature. If a less heat is given to this reaction, only potassium chlorate will be melted, but no oxygen is given out. So, for this purpose, a little black manganese oxide is added to the reaction, which will increase the speed of the reaction and a rapid evolution of oxygen will be taken place. So now here manganese oxide is an inorganic catalyst which will speed up the chemical reaction, but at the end of the chemical reaction it will remain unchanged. So the mass of the manganese oxide will remain same. So there are many reactions that can be speed up by the use of a catalyst. So now, what are enzymes? Enzymes are biological catalysts because they are made up of proteins. They alter the rate of chemical reactions without being themselves changed at the end of a chemical reaction. Now here arises a question that why enzymes are known as biological catalysts. Enzymes are known as biological catalysts because they play an important role in controlling a number of reactions that are occurring in a living organism. For example, enzymes play an important role in digestion of food. There are a number of food substances that animals, including us, consume which are insoluble in water and non-diffusible through cell membrane. For example, when we eat food, they contain carbohydrate, proteins, and fats. These are larger molecules which cannot be passed through cell membranes and are insoluble in water. So for that purpose, enzymes act on them and convert them into a smaller substances which are soluble in water and which are diffusible through cell membranes. This whole process is known as digestion of food. So what is digestion of food? Digestion is the process by which large insoluble food molecules are broken down into small soluble and diffusible molecules. For this purpose, there are digestive enzymes which play a very important role in digestion of food. As you can see that enzyme lipase will act on the fat molecule and will convert it into a simpler building unit, cholesterol and fatty acid. In the same manner, protease will act on protein molecules and will convert them to their building units, amino acids. And amylase will act on starch molecules and will convert them into a simpler sugar, which are soluble in water and diffusible through cell membranes. Besides digestion, enzymes as biological catalysts will play important role in different chemical reactions occur within living cells, for example like in photosynthesis, respiration. Other than enzymes also helps in the synthesis of complex substances from the simpler ones. For example, amino acids taken into the cells may be used to build up the cell proteins. For this purpose, cytoplasm produces special enzymes to bring about such reactions. Other than digestion, there are several chemical reactions in which enzymes act to convert or to break down these complex substances into simpler ones. For example, during the oxidation of glucose to release energy and form carbon dioxide and water. As this reaction involves a series of reactions, so several enzymes are involved in this reaction, catalyzing one reaction and acting together with the other to bring about the complete process. In another example, we can see that sometimes during the chemical reactions, poisonous hydrogen peroxide is produced, which is very poisonous for the tissue. So the enzyme catalase will catalyze the hydrogen peroxide into water and oxygen, which will reduce its poisonous effect. This enzyme is present in plants and animals, but it is usually absent in mammals. But students, you should remember that enzymes are produced only when they are needed. For example, for the process of digestion, digestive enzymes will only produced when there is food to digest in the gut. So these are the some examples which shows that enzymes are biological catalysts. So the next topic is classification of enzymes. There are different kinds of enzymes. Enzymes that catalyze hydrolytic reactions are called hydrolysis. What is hydrolysis? Hydrolysis is the breakdown of complex molecules into the simpler ones with the help of water molecules. As you can see from the name, hydro means water and lysis means splitting. So splitting of complex molecules into simpler ones with the help of water molecules, that process is called hydrolysis. And those enzymes that catalyze hydrolytic reactions are called hydrolysis. Now this is the table which represents different types of hydrolysis enzymes. First one is carbohydrases. Salivary and pancreatic amylase will act on starch molecules and will convert them into the simpler ones. Cellulase enzyme, which is produced by some bacteria, will act on cellulose molecules. In the same manner, there are different proteases like pepsin, which is produced in the stomach, will act on the proteins present in the stomach. Trypsin enzyme, which will be produced in inactive form, trypsinogen, by the pancreas, will act on the proteins. Erepsin enzyme, produced by intestinal gland, will act on the polypeptides molecules. Similarly, lipases, pancreatic and intestinal lipases, will act on fat molecules. There are certain properties of enzymes like catalytic property, reversibility, specificity, heat sensitivity, and pH sensitivity. Now there are certain other characteristics of enzymes. As I have told you earlier, that enzymes speed up or alter the speed of a chemical reaction. Enzymes are needed in small amount in a chemical reaction, and that small amount of enzyme can bring about a large amount of chemical reaction. Same enzyme can be used again and again, and enzymes remain unchanged at the end of a chemical reaction. One major property of enzyme is that enzymes are specific in their nature. How they are specific? They are specific in this manner that only specific enzyme will act on a specific chemical reaction or molecule. For example, amylase will only act on starch, not on proteins and fats. In the same manner, proteases will act only on proteins, and lipases will act just on fats, not on proteins and carbohydrates. Similarly, enzymes are very highly specific catalysts. It means that each enzyme only speeds up a specific reaction. Now you can see from the picture that the molecule that is working with enzyme, that is called a substrate, and the region where it is binding with an enzyme, that region is called active site. So now the role of enzymes in lock and key hypothesis. What was the lock and key hypothesis? It was proposed by German chemist Emil Fischer in 1894. According to him, enzymes have specific shape that directly correlates to the shape of a substrate. Basically, substrate fit into an enzyme the way a key fits into a lock. As we all know, a specific key fits into a lock. Not every key unlock a lock. Similarly, enzymes only allow binding of those molecules that fit into their active sites. We can say that these active sites are the locks that are specific, and few molecules that act as keys can bind them. This whole model of enzyme working is known as lock and key mechanism. From these pictures, it will be more clear to all of you about lock and key hypothesis. In the first picture, you can see on the left side that a specific key, which you can say a substrate, will fit or bind with the specific lock, that is enzyme, and will form a lock and key complex. On the right side of the same picture, you can see that a substrate is binding with a specific enzyme on a region called active site and is forming an enzyme substrate complex. So a specific substrate which contains the same shape as enzyme will bind with the enzyme and will form a enzyme substrate complex. As a result, new products are formed. Then these products are released and the reaction will be completed. Hope all the things are clear to all of you. Thank you. Allah Hafiz.